Hello everyone, wishing you all a lovely day. So we are in chapter 8 where we are having the quick revision of this chapter 8, cell the basic unit of life. Today let us continue with the cell membrane. We all know that the detailed structure of cell membrane was studied after the invention of the electron microscope that was invented during 1950s. So after the 1950s only the detailed study of the cell membrane was made. And the first detailed study was regarding the human RBCs, human red blood cells. And we all know that cell membrane is composed of lipids and they are arranged in bilayer. And these lipids are arranged within the membrane. How it is arranged? The polar head towards the outside and the non-polar saturated hydrocarbons towards the inner side. Okay, the polar heads towards the outside. And the main component of the lipid is phosphoglycerides. And we all know that it is cell membrane also possesses the proteins as well as the carbohydrates. So it possesses proteins and carbohydrates. It is said that approximately 52% of protein is in the erythrocyte and 40% of lipids are there in the erythrocyte. It is accurate and approximate amount of the RBCs and the erythrocyte, the proteins as well as the lipid is being. And on the ease of extraction, we know that integral portion, they are divided into integral as well as the peripheral region. Peripheral proteins lie on the surface and integral proteins will be buried in the membrane. So it is on the ease of extraction it is being divided, classified into two, integral as well as peripheral. Integral means it is buried inside and peripheral means the proteins are seen in the outer surface. As you can see here in the image, it is clearly shown here. So the protein that it is in the peripheral region, that is mentioned as peripheral protein, and the protein that which is within the membrane, inside the membrane, integrated together, that is mentioned as integrated or integral. Okay, integral proteins. Here lipid bilayer we can see, and the yellow color, the yellowish structure that we can see is the lipid bilayers, and the sugar we can see in the peripheral region. So this is the fluid mosaic model of plasma membrane. So who has proposed this fluid mosaic model? Nicholson and Singer in 1972. Cell membrane, this fluid mosaic model of cell membrane was proposed by Nicholson and Singer. So what they in the year 1972. So what they have told is that due to the calcified fluid nature, what is calcified fluid nature? That is the liquid nature. Due to its liquid nature, the lateral movement of the proteins within the overall bilayer, and due to its fluidity also, it was able to move within. Okay, that was the study made by due to its liquid nature, quasi-fluid nature, it is able, the lateral movement was possible, the fluidity was possible. And the fluid nature of the membrane, due to its fluid nature of the membrane, there was certain benefits also. What were the benefits of this one? Cell growth, formation of the intercellular junctions, secretion, endocytosis, cell division, etc. was the benefits due to its fluidity of the nature. And one of the most important function of the plasma membrane, what is it? We know that is the transport of molecules, transport of ions from one place to another place. And with passive transport, we know that without any requirement of energy, when it is moved, that is passive transport. And in the case of osmosis, we have learned that water diffusion taking place, that is the water movement of water by diffusion, that is termed as osmosis. And the polar molecules cannot pass through the non-polar bilayer, then they require a carrier protein. And we have learned about active transport, that is sodium and potassium ions pumps are being used here in the active transport. Next, moving towards the cell wall. We all know that cell wall is non-living and it is rigid in structure. It is non-living and it is rigid in structure. And we all know that it is the identification of the plants. So plants possess cell wall. Animals doesn't have cell wall. Fungi also having cell wall, but it is made up of chitin. We have learned all these things. And what is the function of the cell wall? The main function of the cell wall is that it will be giving shapes to the cell. and also it will be protecting the cell from the mechanical damage. So it will be giving the shape to the cell, and also it will be protecting from the mechanical damage, infections. It will be protecting as such. Then cell to cell interaction is possible. Then it will be acting as, it will be providing barrier to the undesirable macromolecules. And algae are having cell wall. What is it made up of? Algae, they are made up of cellulose, they are made up of galactans, they are made up of mannans and minerals like calcium carbonate. So algae, the cell wall is made up of. While in other plants, what is it made up of? It is made up of cellulose, it is made up of hemicellulose, it is made up of pectins, it is made up of proteins. So the main thing you have to remember here is that regarding the plasmodesmata. What is plasmodesmata? It is the neighboring cells, adjacent cells are glued together, connecting the cell wall and the middle lamellae may be transverse. That means connection. They are the adjacent cells or the connection towards each cells. That is plasmodesmata. Next, moving towards the endomembranous system. So endomembranous system, due to the coordinated, since it is coordinating in function, this endomembrane system, it can be included. Endoplasmic reticulum are included in this one. Then Golgi complex are included in this one. Lysosomes are included in this one. Vacuoles are included in this one. Why? Because the coordinate, since the endomembranous system are coordinating in function, so all these cell organelles, the endomembrane system includes endoplasmic reticulum, they include Golgi complex, they include vacuoles, they include lysosomes. Why the mitochondria, chloroplast, peroxisomes are not included in this one? Because they are not coordinating components. That's what they are not included in the endomembranous system. Next, moving towards the endoplasmic reticulum. What is endoplasmic reticulum? It is a network of reticulum. It's a network of reticulum and a tiny tubular structure that is scattered in the cytoplasm, that is termed as endoplasmic reticulum. You can see here the image of the endoplasmic reticulum. So mainly it is divided into two. What are they? They are two distinct compartments. They are having luminal inside the ear as well as the outside. That means extraluminal, that is in the cytoplasm. So inside as well as outside, it is being divided into two. Mainly the endoplasmic reticulum, those which is bearing ribosomes, it is termed as rough endoplasmic reticulum, and that which is appearing as smooth, those do not possess the ribosomes, it is called as smooth endoplasmic reticulum. So there are two types of endoplasmic reticulum, rough endoplasmic reticulum and smooth endoplasmic reticulum. You can see here in the image the yellow dots that which is seen here, that is the rough endoplasmic reticulum, since ribosomes are present. Here you can see there is no ribosomes present, and these are the smooth endoplasmic reticulum, and they appear to be smooth. This smooth, rough endoplasmic reticulum, what is the function of the ribosomes? Protein synthesis. Main function of the ribosome is that protein synthesis. So we call it as the protein factory, or the protein synthesizing factory. So ribosomes main function of this one is protein synthesis. And here smooth endoplasmic reticulum, what is the main function of that one? Synthesis of lipid. In animal cells and all, we can see that lipid-like steroidal hormones are synthesized by this smooth endoplasmic reticulum. So steroidal hormones in animal cells are synthesized by this. So hope you might have understood the differences between the smooth endoplasmic reticulum and rough endoplasmic reticulum. Now moving towards the Golgi apparatus. Camillus Golgi in 1898, he had discovered or observed, we can say, this densely stained reticular structures just closer to the nucleus. He had observed, and he had named it after his name. That's what it is named as Golgi apparatus or Golgi bodies. Okay, in this one, main important portion is that cisternae. Cisternae, as you can see here, it is stacked parallel to each other, and it is of disc shaped. As you can see here, it is of disc shaped, and this is the cisternae, and these are parallel to each other. It remains as stacks. And here two types of arrangements is there in this one, that is cis and trans. So cis means it is convex, and we can call it as forming phase. It is forming phase, or concave or trans, and it will be the maturing phase. So these are the two types of arrangements. How the cisternae are being arranged if it is as cis and trans. Cis will be convex and trans will be concave. Cis is forming phase and trans will be maturing the phase. So these cis and trans phases, they are entirely different. This cis and trans phases are entirely different, but they are interconnected. They are interconnected. And what is the main function of this Golgi apparatus? The main function of the Golgi apparatus is that packaging of the materials. Okay, the packaging of the materials is the main function of the Golgi apparatus. And this Golgi apparatus, we can say that this is the important site of the formation of glycoproteins as well as glycolipids. The main important portion of the formation site of the glycoproteins as well as the glycolipids. Now moving towards the lysosome. What are lysosomes? They are membrane bound vesicular structures. They are formed by the process of packaging of the Golgi apparatus. They are formed by the packaging of the Golgi apparatus. They are also membrane bound vesicular structures. They also are rich in enzymes. They also have their own enzymes. So they are hydrolytic enzymes such as lipases for the fats and all, proteases for the proteins, carbohydrates for the carbohydrates. Then these enzymes are capable of digesting carbohydrates, proteins, lipids and nucleic acids. These enzymes are capable of digesting also. So that's all about the lysosomes. Now going towards the vacuoles. So vacuoles also we can say that they are membrane bound. Okay, where is it found, these vacuoles? Vacuoles are found in the cytoplasm. What does it contain? It contains water sap, it contains excretory products and other materials that which is not useful for the cells. So those are the things that which is present in the cells, that which is not useful. Here the important thing that you have to remember is that the vacuole will be bound by a single membrane that is termed as tonoplast. Okay, tonoplast means the vacuole will be bound by a single membrane and that is termed as tonoplast. And we all know that plant cells, almost these vacuoles possess 90% of the part. 90% of the volume of the cell it possesses vacuoles. In plant cells, vacuoles are bigger. In animal cells, we can see small vacuoles are there. Then what is the function of the tonoplast? Tonoplast will be facilitating, okay, transporting of the number of ions they will be transporting from one place to another. That is main function of the tonoplast. And also it will be against the concentration gradient. So these two things you have to remember regarding the tonoplast. Then in amoeba, we all know that contractile vacuole is important for the excretion, and in the protist and all, food vacuoles are very important for engulfing the food. These two terms we have come across in our previous classes also. Now moving towards the mitochondria. We all know that mitochondria is not that easily visible under the microscope. It is not that easily visible under the microscope. So the number of mitochondria per cell varies depending upon the physiological activity. Its number, the number of the mitochondria varies in various cells. Sometimes it will be of different shapes also. It occurs in different shapes. Sometimes it will be of sausage shape, sometimes it will be of cylindrical shape, and each mitochondria will be of double membrane structure. What are the double membrane structure here? You can see here it is divided into outer compartment as well as inner compartment. So it is divided into outer compartment and inner compartment. Inner compartment we can call it as the inner compartment that is called as matrix. The inner compartment we can call it as matrix, and the outer compartment, outer membrane forms. The inner membrane forms the cristae. What is the function of the cristae? That is, the surface area is increased in this one. And each of these outer membrane as well as the inner membrane, they will be possessing their own enzymes. They have their own enzymes. As I have told you, outer compartment they will be they are called matrix. Inner compartment they are called as cristae. Cristae. What is the function of the cristae? They will be increasing the surface area. Then the important thing about the mitochondria is that both the membranes, outer membrane as well as the inner membrane, both are possessing their own enzymes. And here we have heard number of times the mitochondria is called the powerhouse of the cell. Why they are known as the powerhouse of the cell? Because they produce cellular energy in the form of ATP. They will be producing cellular energy in the form of ATP. That's what they are called as powerhouse of the cell. Mitochondria, very often it is asked in the exams and all. So you have to learn that. And this matrix, as I have told you, outer membrane, that is termed as matrix. What does this matrix contains? This also, matrix also will be possessing singular circular DNA molecule. They will be having few RNA molecules will be present, ribosomes of 70S. And also that which is components that which is required for the protein synthesis they are having. And this mitochondria, we know that it is divided by the fission. Now moving towards the plastids. Where do we find these plastids? We do find these plastids in the plant cells as well as in the euglenoids. We can find the plastids in the plant cells as well as in the euglenoids. So there are various types of plastids. They are the plastids are being classified into three. What are they? They are chloroplast, they are chromoplast, and they are leucoplast. So chloroplast, what does it contain? It contains carotenoid, chlorophyll as well as the carotenoid pigments. So mainly the chloroplast contains chlorophyll pigment. Then the chromoplast, what does it contain? It contains carotenoid pigments. And what is leucoplast? They are the colorless plastids. Okay, chromoplast and the chloroplast contains chlorophyll and they used for photosynthesis. Chromoplast, they will be containing carotenoid pigments, and leucoplast, which is a colorless plastids, and they will be storing the nutrients. We know that amyloplast will be storing carbohydrates, while lyoplast will be storing oil, and the aleuroplast will be storing the proteins. And now, so we all know that majority of the chloroplast of the green plants, they are found where this chloroplast is being found, that is found in the mesophyll. Okay, mesophyll cells of the leaves. Chloroplast is found in the mesophyll. What is the shape of this one? It can be of dent shaped, it can be of oval shaped, it can be of spherical shape, and it can be of discoid shape, ribbon shape. It varies. Okay, sizes varies. It is said that chlamydomonas green algae will be having 20 to 40 per cell in the mesophyll. It is said as such. Okay, like mitochondria, this chloroplast also having outer membrane as well as the inner membrane. But the inner membrane is relatively less permeable comparing to the outer membrane. That is the important point you have to remember here. Then the inner membrane of the chloroplast, what is it termed as? The inner membrane of the chloroplast, that is termed as stroma. And we can see here the flattened membranous sacs, that is thylakoids. So thylakoids, as you can see here in the structure, which is arranged in the form of piles of coins. Okay, thylakoids, it is arranged in the piles of coins. We can call it as grana. In the singular form, we call it as granum. Then the stroma of the chloroplast contains enzymes. This stroma will be containing enzymes. For what these enzymes? They are that which is required for the synthesis of the carbohydrates as well as the proteins. These enzymes required for the synthesis of the proteins, the carbohydrates as well as the proteins. And what does it contain? It contains double-stranded circular DNA, and also it contains ribosomes. These chlorophyll pigments are present in the thylakoids. Where these chlorophyll pigments are present? It is present here in the thylakoids, as I have told you, which is arranged in the piles of coins like. Then the ribosomes of the chloroplast are smaller when it is compared then to the cytoplasmic ribosomes. So the ribosomes in the chloroplast are smaller, that is of 70S, while in the cytoplasmic ribosomes it is of 80S. So now moving towards the ribosomes. So ribosomes was first observed under the electron microscope by George Palade. Okay, George Palade had observed these ribosomes as a granular structure in 1953 under the electron microscope. So what is it composed of? It is composed of that he found that it is composed of ribonucleic acid and proteins. Ribosomes are composed of this, and it is not surrounded by any membrane according to his observation. George Palade in 1953 he had observed this granular structure, ribosomes. What is it composed of? Ribonucleic acid as well as proteins. The eukaryotic ribosomes are of 80S, while the prokaryotic ribosomes are of 70S. What does this S stands for? That is for the sedimentation coefficient. What is sedimentation coefficient here? That is the indirect measurement of the density as well as the size. So both these 70S as well as 80S ribosomes, these are composed of two subunits. This is 70S and the 80S. are composed of two subunits, and it is bound by the magnesium. In between is magnesium. Then moving towards the cytoskeleton. What do you understand by the cytoskeleton? We can call it as the framework of. It is the proteinaceous filamentous structure that which is present in the cytoplasm, and it is collectively termed as cytoskeleton. So the cytoskeleton will be forming the framework for the cell. That's what the main. And what are the functions of the cytoskeleton? The main function of the cytoskeleton is that they will be providing mechanical support, they will be providing motility to the cells, they will be maintaining the shape of the cells. So these are the functions of the cytoskeleton. Now moving towards cilia and flagella. Number of times we have learned about the cilia and flagella. What do you mean by cilia and flagella? We all know that it is the hair-like outgrowth of the cell membrane. So hair-like small tiny outgrowth of the cell membrane, and we know that due to cilia and flagella only the movement is occurring. Okay, movement, main causes of the movement is due to the cilia and the flagella. It will be moving like oars, like we know that in the boat and all they use oars. Okay, we call it as in Hindi we call it as पटवारे. So उसी के जैसा वो function करते हैं oars के जैसे. So this causes movement. That's what we have learned about the flagella. And flagella we know that compared to cilia, we have seen in the case of paramecium there are cilia. With the help of cilia they move. So flagella we have seen in the case of euglena. So flagella is comparatively longer than the cilia. It is bigger than the cilia structurally. So it causes a big and we know that cilia and flagella both causes movement. Due to both causes movement. Then prokaryotic bacteria also causes flagella. Eukaryotic bacteria also causes flagella. Both these bacteria causes the flagella, but structurally they are different. Structurally in the prokaryotic bacteria also different, in the eukaryotic bacteria also the flagella is different. So it is viewed, this flagella is viewed under the electron microscope, and they have found that this flagella is covered by a plasma membrane, and their core is mentioned as axoneme. And the core, you can see here, it is the electron microscopic view of the flagella. Here you can see the electron representation, they have drawn it clearly and shown it. So the core is termed as axoneme, and the axoneme usually has 9 pairs of doublets of radially arranged peripheral microtubules. You can see here the 9 pairs of peripheral microtubules here, and it is in the form of 9+2 array arrangements. And these central tubules will be connected by the bridges. What is it termed as? Central sheath. You can call it as central sheath. These two are the interconnection. That is the bridge that which is connected, and it is having 9 radial spokes. You can see here. So since it is having 9 peripheral microtubules, it is having 9 radial spokes too. Then they are also interconnected by the linkers. You can see here they are interconnected by the linkers. Then the cilium and flagellum, it is said that it is arising from the centriole-like structure that is called as basal bodies. So where the cilia and flagella is arising, that is from the basal bodies. It is said that it emerges from the centriole-like structures. Now moving towards the centrosomes as well as the centrioles. So what do you understand by centrosome? We all know that it's an organelle that contains 2 cylindrical structures. What is it called as? It is called as centrioles. 2 cylindrical structures called as centrioles. We know that they are surrounded by the amorphous pericentriolar materials. Amorphous means separate. And the centrosome will be lying perpendicular to each other. They will be lying perpendicular to each other. And how they will be organizing? They will be organizing like the cartwheel. We have seen in the bullock cart and all, like cartwheel it will be organizing. And they are made up of tubule in 9 tubule structure, and they are composed of proteinaceous. Central part is termed as proteinaceous, and it is called as hub region. Then the peripheral triplets, that is termed as spoke. Then the centrioles, that is forming from the basal body of cilia and flagella, and spindle fibers that will be giving rise to the spindle apparatus during the cell division in the animal cells. During the cell division only it will be forming, that we have learned. So let us learn about the nucleus. So what is nucleus? We know that it is a cell organelle. So it was first described by Robert Brown in 1831. So 1831 Robert Brown has discovered the cell organelle nucleus. First it was described by Robert Brown, and later on when it was stained in the basic dyes, it was given, this nucleus was given the name chromatin by Fleming, a scientist, Fleming. Afterwards it was stained, this nucleus, this cell organelle was stained in the basic dye, and afterwards he termed it as chromatin. That was given by the scientist Fleming. So this interphase nucleus, when it is not divided, it is termed as interphase nucleus, and they will be having highly extended and elaborate nucleoprotein fibers called chromatin. So what is the structure? They are of spherical bodies and called nucleoli. Singular form we can call it as nucleolus, and they are having the parallel membranes. Within the parallel membranes there is space. The space between this is termed as perinuclear space. The space between the parallel membranes, that is termed as perinuclear space. Then here the outer membrane usually remains continuous with the endoplasmic reticulum. Most probably the outer membrane, you can see here the structure. The outer membrane usually remains continuous with the endoplasmic reticulum and also bearing ribosomes. We know that what is the function of the ribosome? Protein synthesis. It is protein synthesis. And there are some cell organelles that is possessing one nucleus, more than one nucleus. There are some organelles that which is not having the nucleus. Okay, so here even that nucleus, what are the examples for that? Erythrocytes and many mammals and sieve tubes of some of the vascular plants, they don't possess the nucleus. So there are some examples that which is lacking the nucleus also. Here the important point you have to remember is that the nuclear matrix or the nucleoplasm contains nucleolus and chromatin. So this is the important point at which we have to learn here. The nuclear matrix or the nucleoplasm contains nucleolus and chromatin. They are also spherical in structure, and it is not a membrane bound structure, but they are actively, this is the site of active ribosomal RNA synthesis. So we know that protein, they are synthesizing the protein. Ribosomes are synthesizing the protein. That's important point that you have to remember here. So later on, so they in the different stages of the cell division, this is slowly changing into, it shows different structures. So chromosomes, in place of the nucleus, they call it as chromosomes. And this chromatin contains what? DNA and some basic proteins called histones, as well as non-histone proteins, and also RNA. This chromosomes contains, during the different stages of cell division, cells show structures such as chromosomes in place of the nucleus. So in place of the nucleus, it will be showing chromosomes. What does it contain? It contains chromatins, it contains DNA, it contains proteins, it contains RNA. Okay. So every chromosome essentially has the primary construction termed as centromere. So what is this centromere, or what do we call it as kinetochore? So on the each side of the chromosomes there will be disc-shaped structure, as you see here in the image. This disc-shaped structure that is termed as kinetochores. So we can classify these chromosomes in 4 types. What are the 4 types of chromosomes here? We can classify into metacentric, submetacentric, telocentric and acrocentric. So in the metacentric we can see the chromosomes, they will be having, here in the image it is clearly shown. In the metacentric, the centromere will be in the center part and 2 equal arms we can see. This is the metacentric stage of chromosomes. And submetacentric means little away from the middle. So it is exactly middle, centromere is found, here the centromere is found little away from the center part. And the next one is that this is metacentric, submetacentric, and this is acrocentric. Acrocentric means towards the end of its stage we can see, acrocentric, situated at the close of its end. And the last one is of telocentric. So chromosomes are classified into 4 types. Telocentric means that is in the terminal end. Centromeres we can see in the terminal end. So based on the chromosomes can be classified in 4 types, that is metacentric, submetacentric, acrocentric and telocentric. As I have told you, in the metacentric we can see the centromere in the middle portion, and in the submetacentric little away from the center, and in the case of acrocentric we can see it in the close to its end, and in the telocentric we can see at the terminal end. So we can see here the image of the types of chromosome based on the position of the chromosomes. So based on the position of the centromeres, we can see here the image. So this is of metacentric, submetacentric, acrocentric and telocentric. And there are sometimes a few chromosomes they will be having non-staining secondary constriction in constant location, that is termed as satellite. Just a satellite-like appearance it will be having. So that is termed as satellite. And next, moving towards the microbodies. So what do you mean by microbodies? They are also membrane-bound minute vesicles. Microbodies means they are membrane-bound minute vesicles. That's what they are called as microbodies. And what does it contain? It contains various enzymes, and these will be present. These microbodies are present in the plant cells as well as in the animal cells. Okay, it is possessed both. It will be having in the plant cells as well as in the animal cells. So hope you might have understood the concept of microbodies. So microbodies means they are membrane-bound minute vesicles. That is termed as microbodies, and it contains enzymes, and it is present in the plant cells as well as in the animal cells. So we have completed the quick revision of this chapter. Please go through the NCERT text, make notes, prepare notes over it. Happy learning to you all. Thank you for listening me. May God bless you all. Thank you.